Our final speaker today is Nathaniel Gaut. Nathaniel received his undergraduate degree in biology from the University of California, San Diego. He's currently working towards his PhD at the University of Minnesota, Twin Cities, under the guidance of Dr. Kate Adamala. There, he is applying biotechnological techniques to develop synthetic cell technologies. Thank you for joining us. Go ahead, Nathaniel.
Sweet. Thank you, everyone, for being here. Again, my name's Nathan Gaut. I'm a grad student in Dr. Kate Adamala and Aaron Englehart's lab, and I'm going to talk about differentiation of pluripotent synthetic minimal cells via genetic circuits and programmable mating. And so, first off, so synthetic cell technologies is a cornerstone in the Adamala lab, and there's a few ways to define it and work with things called synthetic cells. The way we use them are, they're basically liposomal bioreactors. So something encapsulated with a phospholipid bilayer, so it is distinguished from itself from the environment. And inside we put a cell-free protein expression system, and there's several different ways this could look. So the pure system, or DXPL if you're familiar with those. But basically they're just in vitro systems that are used to transcribe RNA from a gene and then translate that into protein. And so in that sense, we have some encapsulated system that can reconstitute that central dogma of biology. So like the fundamental process of expressing a protein from a gene. And so on top of that, you can start building other new cool technologies that can better mimic other things cells do. And that's the idea of building a synthetic cell, is to try to make it more cell-like, so you can have a better model than in vitro systems, so something more biologically relevant, while being much more simple than a live cell. So it's a lot easier to understand all the different interactions and things happening inside this system. And so obviously, as things become more complicated and you start even coming close to approaching the complexity of a live cell, you're going to need a lot of different ways to regulate all these other pathways and protein networks. And so that's where genetic circuits come in, and they're basically synthetically built means to regulate a pathway. It's something that, as a researcher, you can program and have it do what you want. And a lot of times they're built in blocks, so you can mix and match different things to turn on different pathways. And oftentimes these are built like classical regulatory networks, so they use promoters and activators, repressors, things like that to control whether a gene is on or off. And so the project I'm going to present is an idea of kind of a unique way of controlling a genetic circuit. And it's built on this technology here. So fusing synthetic cells using nucleic acids. And the way it works is you have a single-stranded oligo, DNA, anchored in the membrane of a liposome or synthetic cell. And you have a complement also anchored in a different population's membrane. And what happens is, if they're complementary, they will hybridize. And when they do, they'll pull the two membranes close enough together that they fuse and the contents mix. And so this is really similar to snare proteins, if you're familiar with those. They're membrane proteins that will clasp each other and very similarly pull the two membranes together to fuse. The difference being that snare proteins are not super orthogonal or programmable, in that different snare proteins will fuse with other snare proteins. Now the difference is nucleic acids are one of the most highly programmable molecules there are. You can have dozens of different sequences that will only hybridize with their complement. And so the idea is you can build a much more complex fusion network where different populations will fuse with populations you choose for them to fuse with and not with others. So that's kind of the big idea here, but I'm getting ahead of myself. First off, does this work? Can you build a super simple circuit with this technology? And so we did that, where we have P7 RNA polymerase under the expression of the endogenous promoter. So this just gets expressed by itself, and then in one population of liposomes. And in the other, we have GFP under the control of that P7 RNA polymerase. So this can't be expressed unless T7 RNA polymerase is present. So the idea is this circuit is not complete unless these contents get mixed. And so we put different fusion tags anchored in each of these populations, where big A fuses with little a, big B with little b, and so forth. And then just measured when you put them together, do they fuse? And does it complete that circuit, and do you get fluorescence? And sure enough, big A tagged liposomes only produce fluorescence when incubated with the complement little a. And you don't see any signal when there's different fusion tags that aren't that don't work together. So sure enough, this works. We can build a super simple circuit where you have two parts that are off, and when they come together and fuse, you complete that circuit and it turns on and you get signal. But the whole idea was we're using nucleic acids so we can build much more complex circuits. And you could have different populations that will only fuse with the central liposome, and you won't have them fuse with each other and produce junk that doesn't make that complete circuit at the end. And so we sought out to do this. And this, we use firefly luciferase, which you can have just as a one gene pathway where you have the gene that makes it and you see chemiluminescence. But we artificially split it up into a 10 gene pathway. So this looks complicated, but we just split the firefly luciferase gene up. We have some recombinases that are necessary to process the DNA so it can be expressed properly. And we have the same RNA polymerase that's split, so you need both of these to get it so you can express stuff downstream. We basically just made it so you need every single one of these pieces in this, this pathway to get the firefly luciferase expression. And what we did was we put each one inside a different population of liposomes and had them programmed to fuse to this central one. And so the idea is only if all of these liposome populations are present and fused together will you get that expression at the end. And sure enough, when you have all of them present and programmed to fuse in the correct way, you get a signal. And when you omit just one of the different populations, you see a decrease in signal. So this pathway, this was our pilot, or proof of concept that you can use this this fusion technology to build genetic circuits that are controlled not by genetic regulatory elements, but by these spatial fusion events that are programmed by you, the researcher. And so that's kind of the foundation for this project. And then we just kind of had fun and we're thinking, well, what cool ways can we apply this to synthetic cells and build new technologies? And so we thought about using it to show differentiation or to model it. And so obviously this isn't anywhere close to live cell differentiation, but it's a very simple model that kind of leads synthetic cells into that realm of being able to take a stimuli, change itself, its fundamental characteristics, so it responds differently later on. And so an example of this is what we did, where we took a synthetic cell population and by treating it with a small molecule, you can induce expression of different recombinases. And so this is our differentiation step, this expression of the recombinases. And then we have it fuse with a reporter genome. And this reporter genome is constructed such that there's a stop codon in front of several different fluorescent proteins. And so it doesn't express anything by itself, but if there's one of these three recombinases present, it'll take out the stop codon and or one of these fluorescent proteins. So if you have the three recombinase present, it'll just take out that stop codon, you get expression of M venus, and then it stops. If you have the Dre recombinase present, it'll take out the stop codon and M venus, and so forth. So different recombinases will alter this reporter genome, so you get different fluorescent output later on. And so it worked. So we added the small molecule, had it fused with the reporter genome, and you get expression as you would expect based on that first small molecule step. And we also made the point that this is irreversible. So if you add a second small molecule after the first one, you don't see it change that second small molecule. It keeps that identity from the first one. So adding a rabbit nose after the first small molecule doesn't change what your output later on will be. And so this is like a genetic circuit programmed by the researcher by adding the small molecule that differentiates this synthetic cell. It changes how it's going to respond to this stimulus later on, this fusion event. And you get a completely different phenotype depending on its previous event of adding that small molecule. And so just to show the versatility of this, or the variety, we thought up another way to differentiate these synthetic cells. And instead of adding a small molecule, decided, well, could we do it by fusion events or these mating events? And so to show this, we made use of this biosine pathway. It's a very well characterized pathway where you have different enzymes processing the small molecule, and you end up with a different intermediate depending on what enzymes are present. So if you don't have BioD, you won't be able to get these ones. And we picked this because these compounds are very easy to see on HPLC, so you can get a really good readout on which of these are present. And so this is the experiment scheme where we have this generation 1 liposome, has these 2 biosine pathway enzymes in it, bio A and B. And then depending on which population we allow it to fuse with, or we're calling it mating here, so whichever one it can mate with will change its propensity later on. So it'll have the same enzymes present in it, so A, B, and E, but what this fusion event introduces is these different fusion tags. So this on the left will have big C introduced in the membrane, and the one on the right will have big D, and the one in the middle will have both. Such that later on, they will only be able to fuse with certain populations of liposomes. And so this one on the left will only fuse with this liposome containing bio C, and the one on the right only with D, and the one in the middle both. And what happens then is you have a different enzyme composition or genome inside this synthetic cell, and it's controlled based on this differentiation event up here. So the cool thing is you have a synthetic cell that is mated, and then it has different fusion tags that will allow it to fuse with other ones later on. So it has the same genotype, but is still this differentiated synthetic cell. And so when you have this, so this is the one on the left here. So you're missing that BioD enzyme. So this is the small molecule makeup of this synthetic cell, where you're missing this one. So you see primarily this blue DV small molecule. And we did it with all the other combinations, and you see a different small molecule makeup in each of these. So this differentiation step isn't changing any of the genome or rearranging DNA, but it ends up influencing how that how the makeup of the synthetic cell will look later on based on what it can fuse with. And so, of course, this is a proof of concept, but you can imagine this has implications in metabolism, having small metabolites being made or not made depending on which differentiation step happens, or for signaling molecules. If there's different populations present, they can make different signaling molecules. And so that's where this project is now. So we showed a proof of concept where we're using synthetic cells to construct genetic circuits. And then we showed, sorry, there's a chat, I'll answer it at the end. And then we showed that you can differentiate these synthetic cells via small molecules or by these fusion or mating events. And so future directions for this are, I mean, obviously it'd be really cool to have, to take each of these a step further, to do what I was talking about and make a signaling molecule in these that can influence different synthetic cell populations down the line. But for now, this is where we took it, and it's we're looking to get it published soon, so be on the lookout for it. So with that, just want to thank everyone in my lab, especially Kate Adamala and Aaron Engelhardt for being awesome mentors. and Joe, Hiley, and Brock Cash, who are co-authors with me on this project. And awesome, and thank you all for listening to me.